Yeah, thank you very much.
Yeah, okay. Can everybody hear me? Yeah, okay. All right. Thank you so much for having me here to speak about some of our work. And Yuri, thank you so much for putting this wonderful session together. So I'm going to talk today about our efforts to understand autoimmune disease risk alleles, and specifically our efforts using single-cell data to understand how risk alleles and autoimmunity work. So my talk is entitled "Autoimmune Disease Risk Alleles Affect Gene Expression Dynamically Across T-cell States." I've been studying autoimmune diseases for a while, and sort of my starting point was the recognition that a lot of these diseases have a genetic basis to them. So rheumatoid arthritis, which is a disease that affects multiple joints, especially the small joints of the hand, has a prevalence in the general population of about 0.5%. However, if you have an affected first-degree relative, that risk goes up 3 to 4-fold. And as you can see, if you have an affected twin, it goes up even more, suggesting that familiality and likely genetics is playing a role there. Another disease in the space of autoimmunity is multiple sclerosis. So as you can see, again, the numbers are similar here in that, individuals who are affected with multiple sclerosis are much more likely to have multiple sclerosis if they have a first-degree relative or a monozygotic twin. With a monozygotic twin, that risk goes up 20-fold over the general population. The genetic basis has been elucidated now with genome-wide association studies. In rheumatoid arthritis, this is the 2014 study led by a Japanese team and includes data from European and American cohorts as well. At the time that we put this out, there was 100 risk alleles for RA. We now have a paper in press at Nature Genetics showing 150 risk alleles for rheumatoid arthritis using data from populations throughout the world. And there's been several, major takeaways, major lessons that we've learned from looking at the alleles that have emerged from these studies in autoimmune diseases. So the first, one of the really big lessons is that T-cell biology is strongly implicated in most autoimmune diseases. So first of all, the HLA locus plays a major role in many of these diseases, and the HLA, of course, is intrinsic to the presentation of antigens to the T-cell receptor. So the role of the HLA really does point to T-cell biology. And now looking outside of the HLA, we see that about 10% of the alleles are coding alleles, and 90% are non-coding. So not changing any kind of amino acid position in a site. But those alleles are disproportionately close or within T-cell genes or are in regulatory regions that are T-cell specific. Possible mechanism to think about here is that maybe there's a promoter or an enhancer or something like that that's sitting around a gene, and there's a SNP within that regulatory element, and that SNP prevents efficient binding of a transcription factor or some other regulatory machinery to that enhancer, and that somehow affects the regulation of the downstream gene. With that mechanism in mind, Gosha Trinka and Cynthia Sandro from my group started looking at enrichment of regulatory element markers, histone markers, across autoimmune disease SNPs. This is the result for RA. And what you see is that there's a substantial enrichment for CD4 T-cell subsets. That is, regulatory elements that are specific for CD4 T-cell subsets are disproportionately overlapping rheumatoid arthritis risk alleles. So that was an important clue. And right around that next year, Kyle Farr and Alex Marson published a paper looking at a broad range of autoimmune diseases, taking a similar approach, looking at histone marks. And again, you can see the results are generally very similar, that if you look at autoimmune diseases in general and T-cell subsets in general, that there's this like profound enrichment. So if this is true, what you would expect is that the presence of a SNP in an enhancer or a regulatory element might alter the expression of a gene. And here's an example where that happens. This is a RA risk allele near the BLK gene. It actually sits in the BLK promoter. And what you can see is as your dosage increases, as you go from GG to GC to CC, that the expression of the gene goes down in general, and it's more profound on the red, which is activated T cells, than it is in blue, which is resting T cells. So in general, though, the SNP is having an effect on gene regulation and expression. And sometimes, if you're really lucky, the story comes together very nicely, and the eQTL, that expression quantitative trait locus that I showed you, might co-localize with disease alleles. So at the top here, I've got all those SNPs, including that SNP I just talked about, and the association with gene expression around BLK. And when you condition on the lead SNP, the SNP that's most strongly associated with rheumatoid arthritis, it actually explains all the signals. So this is a case where the same genetic signal that's conferring risk of autoimmunity is also the same genetic signal that's affecting BLK expression. So that's a really happy situation. But most risk alleles don't alter gene expression. So many autoimmune risk alleles, about 50% of them, are within T cell regulatory regions, but only about 20% of eQTLs co-localize with T cell eQTL. So there's quite a large number of SNPs where we don't see an effect on expression, but you do see that the SNP is in a regulatory element, thinking that there must be an effect on gene regulation, but we're somehow not seeing that when we actually look in T cell data. And I just want to give a quick example. This is the CD28 CTLA4 allele. This is the SNP, and we have some functional assays here. On the left, you see the EMSA assay, and what you see on the left is that that when you have the C allele, you see this one specific band, and that specific band indicates that individuals with the C allele are binding nuclear extract or some sort of protein complex that the A allele doesn't bind. And on the right, you see a luciferase assay where you see that both a oligo with the A and C allele in JERCAT T cell lines has enhancer activity, but the C allele has more enhancer activity. And indeed, when you look in high C data, you see that region that SNP is in actually communicates to CTLA4 promoter, again, suggesting a very provocative mechanism that the SNP might affect CTLA4 expression somehow, but it's not an eQTL. So unlike that last example where the SNP has a clear effect on expression, this SNP has no effect on expression that we've been able to see. So that got us to think, what might be going on here? And one possibility is that maybe these alleles have effects on expression that are specific to particular T cell states. So does the functional state matter? So it's possible that the effect of a SNP on gene expression is state dependent. So T cells, of course, take on functional states. They start as naive cells and become memory cells. And then those memory cells can take on different effector phenotypes. For example, they can be Th17 T cells that secrete IL-17 when stimulated, Th2 or Th1, which secrete interferon gamma when stimulated. And of course, there's a range of mixed states. These states are not nearly as discrete as we think they are. And maybe these regulatory elements are only active in certain substates, the substates that are somehow playing a key role in the pathogenesis of the disease in an early stage. So, and understanding how these alleles work, which cell states they work in, and what genes they act on is essential for us to understand disease mechanism and develop drugs. So proof of principle. So this was a study done by Maria Gutierrez-Arcelas and Yuri Baglienko. So it was a sort of a simple experiment. What we did was we sorted memory T cells from 24 individuals, and we stimulated them ex vivo and applied RNA-seq. And then what we tried to do was to identify genes for where there was allele-specific expression, and the allele-specific expression changes as you stimulate these T cells. And then, of course, we wanted to see if these genes were more likely or less likely to be playing a role in autoimmunity. So this is the experiment. So we have 24 individuals here, and again, we've sorted those memory CD4 T cells, and now we're stimulating them. And what we're looking for are these kinds of patterns where we're looking for dynamic allele-specific expression. So the idea is you have a gene, and maybe there's a SNP in the gene, so you can see that some transcripts have the A SNP and others have the B SNP, or the C and the T, I guess, the A allele or the B allele. And then with stimulation, what you might see is that in the beginning, you're sort of even, both alleles are equally expressed, but with stimulation, the gene regulatory situation is changing, and the T transcript is much more abundant. So here's an example of where we see that. This is UBASH3A. This is actually an autoimmune disease gene. When you stimulate for 72 hours, you see that the expression is changing. It starts high, goes down, and ends up somewhere in the middle at 72 hours. And now we look at and see a SNP within that transcript in our heterozygote individuals. And now we're able to tell which transcripts are the reference transcript and which ones is the alternate transcript. And what you can see is that in the beginning, the alternate transcript is really lowly expressed, but it goes up and then goes back down in 72 hours. And in fact, if you look at the ratio of the reference versus the alternate SNP, you can see in the beginning, most of the expression is coming from the reference SNP. As you get into 4 hours, you can see that it sort of balances out. So the alleles have different advantages as you go through T cell activation. And indeed, when we looked at 561 events that were significant to have dynamic allele-specific expression, we found 186 genes, including HLA, CXCR5, and other compelling autoimmune disease genes. And in fact, when we started looking at different autoimmune diseases, we saw that yes, eQTLs in general were enriched for autoimmune diseases, but these dynamic ASE events were even more enriched. So these genes whose regulation is changing with the T cell state. So just to summarize, before I go on to the rest of the talk, so ex vivo activation demonstrates altered gene regulation in T cells, but we recognize that these ex vivo activation states may not represent what's actually occurring in tissue inflammation or in T cells in vivo. So that brought us to the idea that we need to interrogate T cell gene regulation in vivo, which means that sorting cells, stimulating them, et cetera, may not be the ideal way to interrogate the genetics of T cell gene regulation. So the solution that we decided to pursue was to try to map eQTLs at single cell resolution in T cells, taking cell state into consideration. And happily, as we were proceeding with this work, of course, all of you are aware that single cell genomics really has exploded in the last several years, and now it's become possible to do these studies of cohorts looking at 500,000 or 1 million cells. Most of the studies prior to this really have been done in pseudobulk or bulk. So, taking sorted T cells, averaging them, doing RNA-seq, or rather even clustering cells and then averaging the gene expression in clusters. So we sought to actually try to do single cell resolution eQTLs. And the work I'm about to present from here on to the end of the talk is the work from Aparna Nathan and other members of my lab. But she really drove this project, and the goal was to model eQTL effects at single cell resolution. So here you're looking at a UMAP of T cells, and what you're seeing there on the blue on the left is that the SNP is having no effect on gene expression, but on the right, in a limited number of these cell states, in the purple, where you might be plotting the effect of the SNP on expression, you're seeing that there is now an effect on expression in those cell states. So understanding the context-specific T cell regulation. So what we did in this study was we profiled memory T cells from 250 individuals in a Peruvian cohort. We assayed 500,000 memory T cells, and we obtained RNA and surface marker data. And the first question that we needed to ask was, how do you interpret RNA and surface marker data jointly? To this, we used a technique that many of you are familiar with, which is canonical correlation analysis, which is frequently useful for single cell data integration. And in this case, what we had was, for every cell, we had data on gene expression, and for every cell, we had data on surface marker proteins. For T cells, of course, surface marker proteins are so essential to understanding what their phenotypic identity is. And then to put all that data in one space, we used canonical correlation analysis, which maximized the correlation of linear combinations of genes and linear combinations of proteins across cells. And of course, this method goes back to Harold Hotelling in 1936, but has found much popularity in the single cell era. So this was our T cell data. So one way to look at this is you can, apply Louvain clustering and see that there's 31 individual clusters. And you can see, for example, that we have a clear separation of CD4 and CD8 T cells. You also see CD25 regulatory T cells and so forth. But we went back and looked at the canonical variates, those CVs that we use to actually define the clusters, and actually looked at them as individual variables. And what we see is that they actually give us some very important information about the state space that every cell lives in. So every cell, of course, has multiple CVs. CV1 captures that left-right axis, where on the right we have CD8 cytotoxic T cells, so it captures innateness. CD2 captures regulatoriness or Tregness of a cell. CV3 captures effector versus central memory state of a T cell, and so on and so on. And then what we wanted to do is we wanted to have a model where we could use these canonical variates to predict what an eQTL effect was. So we had the typical thing you'd use is to use a simple linear model. We found that our data was so sparse, as in single cell data it is, that we used instead a Poisson mixed model of single cells, where we had fixed effects that captured clinical variables such as age, sex, ancestry PCs. We had fixed effects that captured information about the quality of the data, for example, the number of UMIs or so forth. And very importantly, we had random effects that captured donor and batch. And we're looking for situations as depicted in the bottom, where that cell state interaction with gene expression is what we're looking for.
Okay. Yeah, people are saying the audio cut. I think it should be okay now. Can people? Okay. Yep. I think we're back. Sorry about that.
Can the virtual audience hear me? Back. Yes. Okay. Brilliant.
Okay. You should be able to go ahead.
Great.
Sorry about that.
Yeah, no worries. So here we have a SNP eQTL effect that's seen in CD4 and CD8 T cells. They're colored in red and yellow. And what we've now done is we've reduced the expression in CD8 T cells gradually. So we've created differential expression, but the effect of the SNP on expression is not changed. And what you see is that the Poisson model is smart. It doesn't report a significant dynamic effect ever, but the linear model gets confused by the differential expression, again, because of the sparsity of the data. So we use this model to identify all the genes with an eQTL in our studies. We found 6,500 eQTLs, and then we asked of those 6,500 eQTLs, which ones interact with state, e.g., is there a CV describing T cell state that's interacting with the eQTL effect size? And so this is one example. This is the gene MAF. And what you see is, and in this UMAP, rather than plotting expression, we've plotted the genetic effect of the SNP on the expression of MAF. And what you see is on the left of the plot, the SNP has a very minimal effect on this gene MAF. But on the right, in your most cytotoxic T cells, in red, you see that the SNP has a major effect on expression of this gene. So as I mentioned, we found about 6,500 eQTLs, and 18 and about a third of them have some sort of cell state dependent effect. And when you look at the lead effect, about 2,200 are interacting with cell states. Many of the SNPs have multiple genetic effects, And very interestingly, when you look at the secondary effect, the second SNP that's having an effect on expression, and I'll dive into one of these examples, the majority of those are interacting with T cell state. So this is an example where we have two SNPs that are acting on an individual gene, MDGA1. And what we observed in this and many other examples is that the same cell state may have opposing effects on expression. So there's a primary effect here, which is sitting in the promoter. Many eQTL SNPs sit in promoters. And you have a secondary SNP, which is sitting downstream, probably in an intron, in an enhancer or something along those lines. And if you look at that lead effect, what you see is that in the lower third of CV1, in the least cytotoxic T cells, its eQTL effect isn't as big. And when your cells get more cytotoxic, the eQTL effect gets bigger. But the story is different for the secondary effect. You see the opposite thing, where it has a bigger effect on the least cytotoxic least cytotoxic T cells, but a more modest effect on the most cytotoxic T cells. So you actually have two SNPs that are influencing expression of this gene differently. What about disease alleles? So this is ORM-DL3. This is a rheumatoid arthritis gene. And if you look at the effect of the SNP on this gene, you can see that it has a large effect on granzyme B positive CD8 T cells, but almost no effect on RORC positive Tregs. This is another very interesting gene. This is CTLA4, very important gene for T cell biology. Here, what you see is that the effect on expression is most pronounced in CD4 positive cytotoxic T cells. This is a little bit of a surprise because CTLA4 is not very highly expressed in those T cell states. And indeed, if you look at regulatory T cells down at the bottom, that's where you have the highest CTLA4 expression, but that's where the eQTL is the most modest. So actually, this is a very interesting situation where the SNP has the biggest effect on gene expression when the expression is lowly expressed. These situations, you can imagine that in any kind of aggregation of T cells, either computationally or by sorting, would be missed. So for many state-specific effects, the largest genetic effect is in low-expressed cell states. Our analysis so far is focused on T cell eQTLs that are dynamic. Genes that are eQTLs in low expression states may not be identified as T cell eQTLs at all. The genetic model of disease then might be about cell states with aberrant or ectopic expression of a gene rather than altered high expression of a gene, suggesting that the key disease cell states is distinct from the cell state which we normally attribute to a gene. So there may be a flaw in the logic of saying, oh, this is an interesting gene. It's expressed in this cell state. This must be the driving cell state. In general, we went through and looked at multiple autoimmune diseases. We assessed co-localization, and what we found is that 71% of RA eQTLs are dynamic, and indeed there's a disproportionate enrichment in autoimmune diseases for these eQTLs that behave dynamically across T cell states. In general, all eQTLs are enriched in promoters. This is a well-known thing in the field of eQTLs. But if you start looking at those eQTLs that are sitting in enhancers, those are disproportionately enriched for cell state interacting effects. So some future directions here. So all the work we've presented here is done in blood. So we need to look at tissue data as well. We've demonstrated how T cell eQTLs are dynamic, but are there eQTLs that are only identified in specific cell states? Yes. So as you can see on the right, this is some ongoing work that we have. You see a large effect of this particular SNP on gene expression. So if you look at the effect of a SNP on interaction, you see it here. But if you look at the main effect, it's not a significant SNP at all. So with computational advances and speed-ups, we're investigating all SNPs in all GWAS loci. And of course, functional validation is essential for all this kind of work. So to summarize and conclude, single-cell eQTL studies reveal a broad range of T cell states. These cell states can be modeled with continuous variables, which allow us to define single-cell resolution eQTLs. About a third of eQTLs are dynamic across ex vivo cell states in our study. Dynamic eQTLs are in fact enriched in enhancers. Many are disease alleles. Curiously, some eQTLs have their largest genetic effects in low expression cell states. as we define causal genes and the context in which they cause disease, it is essential to think about SNP effects and gene expression independently. And then a few future directions, we want to apply the same approach to data sets that capture T cell states in human disease tissues. We also want to identify all alleles that are, and all eQTLs, including those that are only seen in restricted cell states. And of course, validation of these effects is important. Oh, I think I lost my acknowledgement slide, didn't make it. So I just want to acknowledge the folks who did the work, especially Aparna Nathan, who did all the single cell eQTL analysis. That data was generated with a large team of individuals, including individuals in Peru who helped us with all the recruitment. It was part of a TB study. Also want to acknowledge Maria Gutierrez Arcelas, who's now running her own lab, and Yuri Baglienko, who did the dynamic allele-specific expression work. And of course, I want to acknowledge my lab. Thank you.
Thank you very much for this great talk. Any questions from the audience, please come to the microphone so that everybody can hear, including the remote participants. I don't think we have questions from the remote participants. Maybe I'll start with a question. I have many, but I'll give you one. Do you know, I mean, it's pretty exciting to me that you can do this. What are the major determinants of your resolution? Is it the number of participants, the sequencing depth, or the number of cells per participant? I mean, let's say if we want to do it again, what should we kind of invest for?
So a great question. So I would say, for any eQTL study, number of individuals is really important, right? So if you weren't looking at interactions, one of the things that we found is you can get away with a fairly small number of cells, as long as you have a lot of individuals. If you're interested in capturing the effect of states, that's when the number of cells becomes important too. So, I guess my answer is both.
Hi, fantastic talk. Seeing eQTL effect sizes on a UMAP is for me. I had a question about the relative. So the concept of deconvoluting bulk RNA to take into account different cell types is well known, and I was wondering whether you've attempted to do the same thing with cell states. If you have a portion of your data set, hypothetically, if you had a portion of your data set single cell sequenced and the rest of it is bulk, and you can use that information to identify likely cell states in the other samples, is that a viable strategy?
I think it is. It's hard. Is sort of my general feeling of deconvolution. Once you get into a specific cell type, we've found it to be generally challenging. I think probably to some extent you can do it. Like you could imagine taking bulk data and I don't know if you might be. I don't know if you can really accurately deconvolute it, but you could imagine finding principal components or something and looking for interactions in principal components. The danger of that approach is that you're almost certainly going to miss really fine scale situations, right? So if you have a eQTL that's really only happening in Th17 T cells and they're single digit percentage of what's in the blood, I think that's going to be really hard to find. CD4 versus CD8, like maybe.
Makes sense. Thank you.
Oh, thank you for the fantastic talk. I really like it. And I think I have two questions. The first question is, have you ever considered applying this approach to potentially clinical data, patients under drug treatment, responder versus non-responder, maybe identify genetic, variants or risk allele that can predict a drug response? Or do you think it's applicable? Yeah. Mm-hmm.
No, I think it's a great question. And I think for us to do that and do it well, we would kind of need clinical trial data. In other contexts, we've tried to look at drug and disease effect. And the problem is, the sickest people are often on the highest doses of the drugs or the most drugs. So our attempts to do this in data in the wild have been sort of confounded by that. So one of the things I would love to see is single cell data from clinical trial participants, where you're really able to separate out the effect of shifting cell states due to disease and shifting gene regulation due to disease versus the introduction in a randomized format of the drug. So, but I think the answer is we would be able to do it with this framework.
Yeah, that's exciting. And another thing, I think I can recall a paper, bioarchive paper recently from Pritchard Lab. It was a hit on Twitter. I think generally their conclusion is there are another explanation why sometimes we couldn't find the colocalization between eQTL and the GWAS is the GWAS SNPs, especially the important ones, are more conserved, but in QTL, the ones that we can identify are less conserved or even could be less important when they can affect gene expression. That's why sometimes we couldn't see enough overlap as we would like, but do you have any comments on that? Or,
yeah. No, I mean, I think it's a thing people have sort of talked about for a while, and the biggest eQTL effects tend to be on I don't want to say the least important genes, but the least conserved genes. And you can imagine, of course, that if a gene is really, really important to the function of your cell, it has to be very well stoichiometrically controlled. But you can still imagine that you may have a SNP that in some very specific circumstance, under the right type of activation, the right kind of stimulation, now suddenly you do see the effect of that SNP on expression. So that's my feeling for why we do see this gap, that I think, if you look in bulk tissue or in GTEx or whatever, you're not going to see for many of these disease genes an effect on expression. But that's, of course, because you're looking at lots of cells under sort of unperturbed circumstances.
Yeah, that's why I do find it's interesting to see many of the cell type specific SNP at the gene regulatory region enhancers. Yeah. Thank you for your answers. Yeah.
First of all, that was a great talk. And this question might be a little bit naive because I'm not much of an immunologist. But it seems like, so you were using site-seq data, it seems, for all of it. So it's the surface markers and RNA. I was just wondering how essential it is for being able to map these eQTLs onto these T-cell states to have that surface marker data. How necessary is that surface marker data as part of this? And I think that was something that I didn't entirely get an understanding of kind of the weights that are making up those canonical correlation vectors.
Yeah, it's actually a great question, and it's not naive at all. And I'll answer your question in two ways. So in a technical way, you don't need canonical variates. Like, you can use the same approach, with RNA PCs, and you'll still find many, but not all of these eQTL interactions. Having said that, where we find the canonical variates really useful and the integration with RNA and protein data very useful is it does allow us to recognize T-cell states, recognize and define T-cell states that sort of fit with the intuition of what we know about T-cell biology, which is important. Now, T-cells are different from other cell types one might be interested in studying, just because there are so many useful protein markers, and probably known protein markers capture the large space of T-cell functionality. So CCA, which is a really an aggressive way to integrate RNA and protein data, like basically you're requiring your RNA signal to be reflected in the protein signal you have, and the protein signal to be reflected in the RNA signal. For T cells, that's a very good strategy. But you could imagine if you're studying something like endothelial cells or even macrophages, where the spectrum of surface marker data is less well elucidated, then the site-seq may not, analyzing site-seq in this way, I should say, may not necessarily be quite as helpful.
Okay, that makes sense. Thank you.
Yeah, maybe I will ask one more question. I think it's related also with the question of somebody in the Q&A session. Maybe we missed what tissue exactly did you do your single cell profiling, and more generally, for example, for rheumatoid arthritis, do you think the results will differ if we look at circulating cells versus, let's say, from the site of the disease, like from the joint?
Yeah, so I mean, I think that's a great question, and it's something we're very interested in doing. So my kind of quick take on it, looking to the future. Well, so first of all the data I presented here is from peripheral blood, which is of course what we're best at getting from people. I think it's really important to look in tissue because, of course, you can imagine that the key cell states that are driving disease might well be present in the tissue, but much more absent in the blood. But my sort of feeling is probably the cell state distribution in tissue and blood is well, we know that it's different. So, for example, T peripheral helper cells, granzyme K positive CD8 T cells, we see that in inflammatory tissue. We see it in blood, but to a much lower extent. So in a way, the question becomes like a little bit about your previous question. It's a little bit about power, right? So if you go to tissue, you'll probably be much better power to find the relevant eQTL states. Maybe you can do a lot of things in blood. Hard to know. Maybe those cell states are there, but they may be in a vanishingly small proportion. So you may need massive cohorts.
Yeah. And not to overcomplicate this, do you think you can add an additional dimension of TCR specificity?
Yeah, that's a great question. So this is something we're thinking a lot about, like what's the link between HLA QTLs and T cells and TCR QTLs and T cells and TCR, and so yeah, I think the answer is yes. Again, it's an active research problem for us, so we haven't quite figured it out, but yeah.